just to review, let's draw what the mitochondria looks like. So what are the parts of the mitochondria? What are the features that are going to become important when we talk about the electron transport chain? Okay, so it has two sets of phospholipid bilayers. It has an outer and an inner membrane. What else? Okay, where's the matrix? Inside the what? Yeah, inside here is the matrix. Okay. What do we call this? The what? Nope, it's not out of the cell. But you're kind of along the lines. It's basically a description of what it is. It's the intermembrane space. Okay? So in between the two membranes. So what else? One last feature that's important about the inner membrane. Well, you're jumping ahead a little bit. Just about the membrane structure itself. It has a lot of folds that we call cristae, right? So it's highly invaginated. Why is that important? Huge surface area. Huge surface area so that we have lots of space for many, many copies of the electron transport chain. So just a small organelle can make a lot of energy. Okay, so these are called cristae. Oh my, this is running out. Okay, so those folds in the inner membrane are the cristae. And as I mentioned, depending on what type of cell it is, like a muscle of the heart, or even that tail fiber of the sperm, there are a lot of mitochondria in some cell types and not so many in others, depending on how much energy the cell types need. Okay, so in addition to these features, you should also know that these invaginations of the inner membrane are called cristae. So this is really key for the final stage of that complete oxidation of now the glucose to the oxygen in the water. So in this inner membrane is where you find the electron transport chain. And the outer membrane has a very different structure. So the inner membrane, it's a phospholipid bilayer, just like the plasma membrane, like we talked about its feature of being selectively permeable. Only certain things can get in and out. Same thing with the outer membrane, but the outer membrane also has protein channels called porins that really allow the free transport of larger molecules like acetyl-CoA to get into, or pyruvate to get into the mitochondria. So these porins are important for the function of the outer membrane. They're not found in the inner membrane. So with all that, those electron carriers that were produced either by glycolysis or then inside the mitochondrial matrix by the citric acid cycle. So the two events that happen, so glycolysis is happening outside of the mitochondria. The pyruvate is then transported across where the transition step, the citric acid cycle, and the electron transport chain or system are found. So in the matrix itself is where the chemical reactions of the transition step, converting pyruvate to acetyl-CoA, and then acetyl-CoA being shuttled into the citric acid cycle occurs. On the inner membrane is where you find the electron transport chain, those three large enzyme complexes that are required for pumping protons out of not the mitochondria, but out into this intermembrane space, creating a proton gradient that can be used to then phosphorylate the energy necessary to phosphorylate ADP to ATP. So we're going to talk about that. So remember that NADH, an electron carrier, but it's also very readily able to donate electrons because they're high energy electrons that they're carrying that are binding that proton to the NAD. And it's because it transitions to an unstable state that with the release of that proton and these two high energy electrons, that then when it's converted to this more stable NAD state, then it drives the release. So the release of the electrons first converts NADH into this kind of unstable intermediate, but then with the change of or rearrangement of the bonds so that you get a double bond between the carbon and the nitrogen, then it's a stable form that can then be recycled. So all of these molecules, rather than just making more NAD, the cell doesn't have to make more. Instead, it's recycled back to the cytosol and also to the matrix within the matrix where it becomes an electron acceptor again. Okay, so in the process of cell respiration, we already talked about that we get 2 ATP from glycolysis. But really the bulk of ATP, 28 more ATP, are made from oxidative phosphorylation. And that's electron carriers like NADH or FADH2, converting this or driving this unfavorable reaction with the help of a key enzyme, ATP synthase, okay, and the electron transport chain. So, as we talked about for the oxidation of glucose, you could just burn glucose and get a lot of release of energy. But the form of the energy that's released is heat, and that's not useful in a cell. So what the cell does is split it up into lots of chemical reactions so that it can harvest that energy. In a similar way, NADH could simply donate its electrons to oxygen directly. But by splitting it up into many steps of electron transfer, the cell is able to, or the mitochondria is able to create an imbalance across the inner mitochondrial membrane that's useful for driving this unfavorable reaction of ADP to ATP. So here in this cartoon, you have the electron transport chain, and then here's represented the ATP synthase, where the protons then re-enter the matrix and drive the phosphorylation of ADP in oxidative phosphorylation. So this first stage is the actual transport of the electrons. That really is what's driving the movement of protons across this lipid bilayer. So remember, you have to kind of remember back to Chapter 11, the rules of what gets across a lipid bilayer freely and what doesn't, and we know ions don't. Anything with charge doesn't. So that by pumping the electrons, or pumping the protons out into this intermembrane space, they're not going to be able to freely cross back. So you create that imbalance. Okay. The proton gradient is then used to drive the production of ATP. So ATP synthase with the energy that's released from the flow or the return of these protons to the matrix is able to catalyze an unfavorable reaction and harness that energy in a useful form, not just as the loss of heat. And it's actually quite efficient because of the protons, the number of protons, or the imbalance that's created across the lipid bilayer. Only about 10% of the energy is lost as heat. So this is what we call chemiosmotic coupling, because it's a way of driving an unfavorable, or coupling an unfavorable reaction with the favorable movement of protons down their gradient from the higher concentration to the lower concentration, which would happen naturally without energy. So remember, passive transport or diffusion occurs down the gradient, active transport the other way against the gradient. And active transport is the one that requires the energy. So the electron transport chain, though, is actually a set of three complexes. So there are many steps in the transport of the electrons that help to pump the protons across this membrane. And the ATP synthase, as we'll learn, is actually has two important parts. It's the part that's a channel for the protons, and also a part that's the enzyme that catalyzes the reaction of ADP to ATP, the phosphorylation of ADP. So here's an electron micrograph of the inner membrane that's studded with these protein complexes that are part of the electron transport chain. So let's take a look closer at what's actually happening. So remember that in cell respiration, we're already talking about what's happening after glycolysis. So after the food molecules have been broken down, and then those subunits have been transported into the cytosol, and the glycolysis in the cytosol has converted the glucose to pyruvate, or from fatty acids that are in the cytosol, they're transported through these porins and then actively transported across the inner membrane to get to the matrix, where they can be converted to acetyl-CoA. Acetyl-CoA is then converted to a 6-carbon molecule and shuttled into the citric acid cycle, so converted to citric acid. And then the products of the electron carriers, FADH2 and NADH, from the citric acid cycle are then shuttled into the electron transport chain. So the proton gradient, so this, the only way that you can get enough energy to catalyze the phosphorylation of ADP is to have an accumulation of protons outside of the matrix, intermembrane space. And so the 3 complexes that help to do this are the NADH dehydrogenase complex, where this is now NADH donating its 2 electrons to the complex, which then results in protons being moved across the membrane. Then ubiquinin passes the electrons to another complex, which in your book they call it the cytochrome reductase complex. And then and more protons as a result of those 2 electrons being transported. And remember, these are kind of high energy electrons that are being transported. And the ubiquinone and the cytochrome C are helping to maintain them in this high energy state. But basically, each step of the way, they're releasing some energy to help pump the protons. So remember, pumps are a form of active transport, so they're really unfavorable. They're going against the concentration gradient. So it's the energy from the electrons, the transport of the electrons, that is helping to move them in this direction from low concentration to high concentration through these protein complexes. And it's basically these steps from NADH dehydrogenase via ubiquinin to the cytochrome C reductase, via cytochrome C to the cytochrome oxidase complex, and then finally to the terminal electron acceptor, the oxygen molecule, that drives that energy that's released by each of these steps, that drives the movement of protons across the lipid bilayer here in the inner mitochondrial membrane. So it's similar to a slinky going down the steps or something. It just keeps on kind of continuing down until it gets to that electron, that terminal electron acceptor. So this process of the movement of the electrons is actually energetically favorable and helps to drive the proton movement. And if you draw it in sort of how we draw our energetic reactions, or the graph out the energy level, so this would be the level of energy, the electrons are moving down. So the delta G is negative or favorable along that pathway. All right, I'm going to stop there and we'll continue next week. What happens when, for example, a carbohydrate comes into the cell, like glucose? Where does it go? What happens to it in the cell? Just to review. Glycolysis, which happens in the cytosol, okay? Then from this arrow on, everything's happening in the mitochondrion, okay? So, so from that 6-carbon sugar being split into 2 3-carbon sugar, 3-carbon molecules, pyruvate, and then converted into acetyl-CoA, which is then becomes another 6-carbon molecule and is cycled through the citric acid cycle. So glycolysis producing a little bit of ATP, a little bit of NADH, but the citric acid cycle, really the bulk of the electron carriers produced there, And then we're at the stage where those electron carriers, NADH, FADH2, are used in the electron transport chain. Okay, now, electrons in the electron carriers are high energy electrons. They're very unstable. And so being that they're unstable, they're easily donated to another molecule. And really this electron transport chain is just a series of steps away. Instead of just passing off the electrons directly to oxygen, which would release a lot of energy in a useless form, heat, the energy is captured in little packets in a useful form by making this process of passing off electrons from the electron carrier to oxygen in several discrete steps. Why is that important? That's important because the energy, so this is written at an angle because each time the electrons are passed to another carrier, it becomes more stable, releasing a little bit of energy. That energy is then used to drive an unfavorable reaction of moving the protons across that inner mitochondrial membrane. So remember, we're inside the two membranes of the mitochondria in the matrix here for the citric acid cycle, and then along that inner membrane for the proton gradient or proton motive force that's created for the electron transport chain, which is all inside that membrane, which is this gray bar behind the boxes in this figure. Okay, so each one of these steps, and there are three major protein complexes that carry out the pumping of the hydrogen ions out as the electrons pass from molecule to molecule. The last one of which holds on tightly to oxygen until it receives those electrons. That's the terminal electron acceptor. And then it's converted rapidly to a harmless form, the product of water. Okay, so if it's not held on tightly, it could wreak havoc in the cell because it's in this very toxic oxygen radical form of superoxide. Okay, so remember, these are the products of cell respiration. And we'll talk about these also later when we talk about these being the starting material of photosynthesis. Okay, so what's really happening at each of these steps? Well, the first complex is NADH dehydrogenase, an enzyme that receives electrons from NADH. And in the process, NADH is reduced to NAD, and then the protons are pumped thanks to a channel protein in this complex that changes conformation with the using the energy of the electrons. So here on this side of the figure is the amount of energy. And each time there's a drop in the amount of energy, that's the amount of energy being released and being used to drive. So there's a couple other things happening here along the membrane. These are all happening in the membrane, the inner membrane of the mitochondria. The electron flow goes from this first NADH dehydrogenase complex to a cytochrome reductase complex, also known as cytochrome BC1 complex, and then to the cytochrome oxidase complex, the one that holds on to the oxygen and converts it to water. There are a couple intermediates that need to carry these high energy electrons to the next stage or the next acceptor. In between the NADH dehydrogenase complex, ubiquinone takes those electrons and then passes them on to the cytochrome reductase complex. And then in between the two cytochrome complexes, cytochrome C carries the electrons. As these electrons are being transferred and the protons are being pumped out, there are a couple of gradients that are occurring or being created along the membrane. So outside of the inner membrane, in that intermembrane space in between the inner membrane and the outer membrane of the mitochondria, there's the accumulation of these protons. Okay, so now you have a gradient, a chemical gradient of protons where it's high in that intermembrane space, low in the matrix. You also have, because of the charge, an electrical gradient, right? Similar to what we call the membrane potential. So there's a membrane potential like what we talked about across the plasma membrane when we talked about the sodium-potassium pump. But in this case, it's the protons that are creating that membrane potential. That potential carries energy, and that energy is then used to phosphorylate ADP. So, as the electrons move downhill, you get useful energy stored. So it's stored in this imbalance between the protons on the outside or in the intermembrane space and in the matrix. And that can be thought of as energy stored really in any kind of energy storing device, even something like a battery. So you can think of the electron transport chain as the battery of the cell, because similar to battery, it's really storing that energy until it needs to be used. Now it's constantly being used by the cell if the cell is alive. So in a battery you have the positive and the negative end, and it's the flow of electrons that help to, if you create a circuit with it, that help to transfer energy, right? And similarly, in the electron, in the cell, the energy can be lost in a kind of useless form as heat energy. So when you have a battery and you just make a circuit, you'll notice that the wires get hot, so there's some heat being lost in that reaction. But you could also make a circuit that drives a pump, and that's really what the electrons are doing in this electron transport chain. So they're pumping protons, not water in this case, but they're doing some useful energy in this, useful work in the cell, thanks to capturing that energy in a useful form. Okay, so once the electrons move, the protons move, they're coupled together. And this work is called chemiosmotic. I was hoping to show doesn't seem to work. Oh, just skips to the next one. During lab, I'll try to get up the animation for this because I think it's useful to watch it. But for now, let's move on. So proton pumps. So now we have the electron transport chain. storing the energy, and that energy is used to drive the proton pumps, which are working against the gradient. So they're serving a purpose of doing uphill work. The proton pumps themselves are active transporters or carrier proteins, and they're active transport because they're going against the gradient. So they're going from an area of low concentration of hydrogen ions to high concentration. They're allosteric in that they have a shape change as part of this process of moving the protons. So first, a proton binds specifically based on its charge and its shape to the transporter. Then there's a conformation change once that proton is bound. And there's a high affinity for protons due to the a nitrogen atom being in that binding site. And then once there's another conformational change that occurs in order to release the proton to the outside of the inner membrane into that intermembrane space. And that happens because there's a loss of the affinity. So there's the conformation wants to return to its from its low affinity to its high affinity conformation. And then it just cycles through again through these three conformations. The A, where it's very high affinity for protons, and then the B, where it changes shape, C, where it opens and becomes a low affinity for the proton, and so the release of the proton is favorable. The part that requires energy, that uses the electron, the energy captured or stored by the coupling to this electron transport, is the conversion of this conformation B to conformation C. And that's where the electron transport chain becomes important. And these types of conformational changes occur at each stage of the electron transport chain. So the NADH dehydrogenase complex, the cytochrome reductase, and the cytochrome oxidase complexes. Okay, so what drives these reactions or these electron transfers is that the electrons are trying to get to a more stable state. And they're very stable, or they're very highly bound or tightly bound to oxygen. So as you go through each of these energy carriers along the electron transport chain, the electrons become higher and higher, more and more stable, and less likely to be given up. So the final stage of the electron transport chain is the complex called the cytochrome oxidase complex. And this is where the terminal electron acceptor is. So just one thing to note, in the beginning of the electron transport chain is NADH dehydrogenase, right? So the starting material for the electron transport chain is NADH. But what do we get from the Krebs cycle or the citric acid acid cycle as far as electron carriers. So what do we get? Do we get just NADH? Yeah. FADH too. Okay, so these are two important products of the citric acid cycle that are the electron carriers that are shuttled into the electrons transport chain. So I've told you what happens to NADH. What happens to FADH too? It still goes into the electron transport chain. What happens is, instead of entering at the NADH dehydrogenase complex, it enters at the next complex, the cytochrome reductase complex. Okay? So what does that mean for how many protons are being pumped as a result of FADH2? Okay, so that's less protons pumped as a result of that skipping that first complex, right? So between the two electron carriers, though, you're getting a lot of protons pumped across that membrane very rapidly. And both of them end with the cytochrome oxidase complex, which is if the respiration is aerobic respiration. If it's not aerobic respiration, again, even with NADH starting at the NADH dehydrogenase complex, it doesn't get to this final cytochrome oxidase complex. And so it results in less protons being transferred and less ATP. Okay? So less protons means less potential energy to be used in phosphorylating ADP to create ATP. So there are some variations that cells have. They can grow, they can carry out anaerobic respiration, but instead of oxygen as the terminal electron acceptor, they're using other electron acceptors like sulfur or nitrogen, nitrates and sulfates, which don't produce quite as much of a proton gradient, and so not quite as much stored energy to be used for producing ATP. So in the ideal circumstances, oxygen is the best for the net yield per glucose molecule of producing the most energy. And oxygen is tightly held by a couple of subunits within this complex. So this multi-subunit complex is made up of protein subunits that are encoded by the genome of the cell, here kind of in this transparent or white part of the protein. And then the colored part are actually encoded by the mitochondrial DNA. And together they form a complex that that accepts electrons and pumps the protons out, as well as holding oxygen tightly until it's oxidized, until it receives those electrons, until it's reduced to water. Okay, so in this subunit, there are a couple of key features. There are metal ions and a heme group, and those help to bind the oxygen tightly as well as transfer the electrons from the cytochrome C. Okay, so the electrons come into this subunit and are transferred to the metal and then to the oxygen. And the oxygen is held by the heme until the protons are brought in to be used in that chemical reaction of protons and oxygen combining to form water, the final product of, or the output of cell respiration. So how many protons do you get pumped? You get 4 protons pumped for every 4 electrons entering. And the 4 electrons accepted by the 2 oxygen molecules are combined with hydrogen to form water. So you get from 1 oxygen molecule 2 water molecules when you add 4 protons. So there are protons being used in this chemical reaction, and there are protons being pumped to the inner membrane space. So when electrons move along these 3 main steps of the electron transport system, a proton gradient is created. The gradient is both a concentration gradient that affects the pH. So the pH being lower when there's more hydrogen ion concentration than the inside in the matrix where there are less hydrogen ions, so it's a little bit higher. It's not quite the difference of pH 7 to 8, but it's more like pH 7.2 to 7.6 or 7.8. It's a small pH difference, but the concentration gradient is an important driving force for phosphorylating ADP. There's also an electrical potential, so just like we learned about the membrane potential across the plasma membrane, same thing in the mitochondria, there's a membrane potential created by the movement of these protons and the imbalance of the protons across the inner mitochondrial membrane. So why is this important? Well, it's really these imbalances that are used to by the ATP synthase to drive the unfavorable reaction of converting ADP to ATP. Okay, so it's the electrochemical energy that used to drive in a mechanical way to drive this ATP synthase. And there are really 3 key parts to this protein. It's a multi-subunited protein. Here's the ribbon and sheet model, but basically the space filling or the diagram here that shows the structure can be divided into really 3 major parts. There's a part that crosses the inner mitochondrial membrane. That's the transmembrane carrier. This is where protons will enter from their area of high concentration. They'll pass through and drive a conformational change that results in the spinning of this neck. And then this globular protein down here, the ATPase, will transfer phosphate to ADP to drive the phosphorylation of ATP, of ADP to ATP. So it really just physically pushes together the inorganic phosphate with the ADP to create that ATP. And it's the energy from the favorable movement of the protons from the area of high concentration to low concentration that drives the movement of the ATP synthase that helps to push these two molecules together and catalyze that reaction. So for every 3 protons that flow through ATP synthase, there's one ATP. One of these chemical reactions occur. And this is really happening quite rapidly. So as long as there's the other stages of the cell respiration are occurring, this will continue to drive ATP synthesis at a rate of about 100 ATP per second. So there's actually 30 ATP that are made in cell respiration from a single glucose. 2 of them are made from glycolysis. Remember, it's actually 4, but because 2 are used, you get a net production of 2. And then the rest are made by oxidative phosphorylation, which is this ATP synthase coupled to the electron transport chain. So the whopping bulk of the energy in the cell is made in the mitochondria by this process. So ATP synthase is not unique to our cells, mammalian cells. It's found in virtually all forms of life. So bacteria have an ATP synthase. They don't have mitochondria, but they have their electron transport chain in their membrane, in their cell membrane. Plants have ATP synthase, which we'll look at in a moment. Algae, fungi, animal cells. All of these have ATP synthase. And I'm not having much luck with the movies today. This should be playing when I hit the arrow. Yeah. Even what? For example, protozoa? Yeah. The only parasitic, they're not usually called organisms, but the only parasites that would inhabit would be viruses, yeah, because they don't have their own metabolism, yeah. Okay, so enough of cell respiration. I'm going to turn the page and move on to photoautotrophs, plants, what goes on in plants. So remember, plants are the bottom of the food chain. They're the ones that can make their own food. So their metabolism is different from ours. We have to ingest food, break it down, use it in cell respiration. Plants, they can use light to produce energy, so they don't need to intake food. They do need to intake water. So what's the general reaction again for photosynthesis? Just to review. What are the starting materials of photosynthesis? Carbon dioxide. What else? Water. Okay, and light, yeah. So, and then what do you get? right? Sugars like glucose and oxygen. So this should look kind of familiar, right? This is just basically cell respiration turned upside down, right? Cell respiration is glucose and oxygen, leading to the final products of CO2 and water, right? So we're just flipping it on its head, turning it around, running it backwards. And in plants have electron transport, but the electron transport happens, they have mitochondria, but the electron transport that's important for photosynthesis happens in chloroplasts. So photosynthesis is occurring in these organelles that we don't find in non-plant material, animal cells. But the interesting thing is that they have a similar structure to mitochondria, having multiple membranes. In this case, they don't have 2 lipid bilayers, they actually have 3. So this gray line and the red line represent the outer membrane and the inner membrane, and then they have an additional set of membranous compartments, the thylakoid, which is also bound by and separated from the stroma by a lipid bilayer. So the stroma is the space in between the inner membrane and the thylakoid membrane. Chloroplasts are also where you find sugars stored, because it's the stroma where the biosynthetic reactions will occur. So sugar production will occur. But inside the thylakoid membrane is where some very important on the membrane itself, where some very important reactions, the light reactions, occur. And it requires, it has to be on the membrane because it's this electron transport process, similar to what we just talked about in cell respiration. Also inside the thylakoid space, now not in the thylakoid membrane, but in the space, which is why these are colored green, are the pigments, the chlorophyll pigments. And those are what help to capture the light energy to begin this light-dependent reactions of photosynthesis. Chlorophylls? Okay, so there are a lot of similarities. We have multiple membranes, but they're also, and also chloroplasts have their own DNA as well. They have ribosomes to carry out the central dogma of DNA to mRNA to protein. Similarly to mitochondria. But they have this additional area of the thylakoid. So let's look and see what happens in photosynthesis. We already learned from our lab experience. We got to see chloroplasts, the first lab that we did when we were looking at the leaves under the microscope. And then more recently, we did the Hill reaction to measure the activity of photosynthesis in chloroplasts isolated from plant cells. So that's without the other parts of the cell. We were just looking at the chloroplast activity. And really what we were measuring is this step 1, right? So we held the chloroplasts up to light, and you could see a change in the absorbance as a result of the reduction or the photosynthetic activity of those chloroplasts. And really what happens in the light-dependent reaction is electron transfer. So what do we have as the electron acceptor in our test tubes? DCIP, right? So it starts out blue, and then as it accepts electrons, it becomes colorless, and then we saw that drop in the absorbance as a result of the change in that color. And then there's also another reaction called the Calvin cycle that happens that doesn't require light. And hopefully from that second cuvette that we tested, where we covered it with foil, you understand that this first reaction really doesn't happen, right? You shouldn't have seen any change in the absorbance in that sample unless there's light. So there's no photosynthesis unless there's light. So the light has photons that can be captured, and thanks to chlorophylls, and those with the presence of water, electrons can be transferred in the thylakoid membranes, converted in. So the waste product here is oxygen, but the useful form, so again, it's a series of breaking down this electron transport into a series of steps so that we can capture the energy in a useful form to do work in the cell. And there are really two photosystems that are important. Photosystem 2 is important for creating ATP in the cell, in the plant cell, and photosystem 1 is important for creating NADPH. Then the Calvin cycle is where the carbon fixation reactions occur. And that's not in the membrane at this point, but in the stroma, in that space in between the thylakoid and the inner membrane of the chloroplast. That results in the production of sugars and other building blocks of the cell. So the light can be captured in the form of high energy electrons. The high energy electrons, again, kind of stepwise are moving from less and less stable to more and more stable states. And with the movement of protons still creating an electrochemical gradient across that thylakoid membrane, that energy battery of now the plant cell can be used then to drive ATP synthesis and NADPH synthesis. So the key molecules that capture that light energy are chlorophylls. There are a wide variety of chlorophylls. And the wavelength of light is in the visible spectrum. But for example, if you have chlorophylls from a green plant, so then the green and yellow light is not, those wavelengths are not absorbed, they're reflected. That's why we see the plant as being green. And it's really the red and the blue light wavelengths that are being absorbed by these chlorophylls. The way that the chlorophylls are situated is they're actually in the membrane. They have a hydrophobic tail, and they're clustered into areas called antennae. As the light comes through the leaf, so there's a thin epithelial layer of cells, but then you have a lot of chloroplasts in the leaf. You have a lot of cells with chloroplasts in the leaf. And then each of these chlorophylls within the chloroplast has a metal ion surrounded by a heme group. That's so in the blue here is where the electrons that are captured from the sunlight are then kind of trapped. And they tend to move, they're still very high energy electrons, so they tend to move and rapidly change the bonds, making the chlorophyll very unstable. So they move from atom to atom in this blue shaded area. The chlorophyll being unstable, it wants to give up its electrons. So they tend to jump from chlorophyll to chlorophyll, but then eventually they come to a reaction center where they're passed on to a special pair of chlorophyll molecules that then split that energy into the electron pairs. Okay, so here it's kind of, this is in, just to orient you, this is the thylakoid membrane. This is the outside, in between the membrane and the inner membrane of the chloroplast, so the stroma area, and this is the inner thylakoid space. So the light gets through to this area of antennae, where lots of chlorophyll molecules are clustered around a reaction center. So the light energy in this process lifts the electrons in the chlorophyll from a lower energy state to a high energy or unstable state. It's passed from molecule to molecule, so there's an electron acceptor that will reduce the level of excitation of that electron, and so it will be favorable for this very unstable state here in the chlorophyll. We'll pass it on to an electron acceptor readily to kind of get the electrons to a more stable, still high energy, but more stable state. In the process, that acceptor molecule is reduced, and the charges separate thanks to those two special electron acceptors in the reaction center. Okay. So the oxidized chlorophyll can then be returned to the charge separation, be returned much more stable. Another electron lost and a donor that's in this reaction center. Now you have the charges are separated. You still have the high energy electron that can be pumped into photosystem 2. And so here, just to orient you, there's photosystem 2, and with the help of water, it's split by an enzyme in this photosystem 2 complex. The electrons are transferred by these intermediates that are in the membrane of the thylakoid. So plastoquinone here in this case in the plants. And thanks to the high energy electrons stepwise losing their energy or going to a lower energy state, they are helping to pump protons against their gradient. So there are three. There's the antenna complex with the reaction center. There's the proton pump, and then the plasticinin, which combines with photosystem 1, and then again we get the transfer or the as a result of the energy stored in released during this electron transfer from photosystem 1, we get the result of the reduction of NADP to NADPH thanks to the enzyme associated with the membrane, ferredoxin NADP reductase. So we're getting basically it's not shown here, but the cytochrome B6F complex here is that proton gradient is used to derive the phosphorylation of ATP. And the movement of the protons, again, just like I talked about with ATP synthase, it's the same enzyme here, where the movement of the protons down their electrochemical gradient is what's generating the ATP. So there are two things happening in the light-dependent reaction. There are two products, important energy-rich products, the electron carrier of NADPH and the energy currency of the cell, the ATP, that are synthesized during the light-dependent reactions. And again, if we graph out these reactions that are on a graph showing the redox potential, you can see that it's, again, a membrane potential created by the transfer of these electrons to the input of water. And those electrons are going from a very excited high energy state stepwise to lower energy states until the photosystem 1, the terminal. And then from there, finally, the NADP is being reduced. Okay, so those electrons are being transferred to create a high energy electron carrier, or ATP. So here in this graph, you can follow the path of the electrons from the water molecule to the terminal product, the NADPH. Okay, so there are two separate things happening here. There has to be an electron acceptor at the end of this procedure. The end of this series of reactions are transfers of electrons, and NADP is that electron acceptor. So step 2 is the Calvin cycle, the part where CO2, the other input of photosynthesis. Okay, so water is important for that light-dependent reaction. CO2 is used in the light-independent reaction, the Calvin cycle. And really what I want you to remember is this key first step, and then in general, it's a cycle similar to the citric acid cycle. So similar to the citric acid cycle, where oxoacetylacetate is recycled during that process, is this rubisphate in this first part, or first step of the reaction, that's combined with oxide thanks to the rubisco enzyme. This is recycled in the Calvin cycle. So each time a new carbon dioxide molecule, how does it get into the chloroplast? How would carbon dioxide get into the chloroplast? It just diffuses across the membrane. Remember, it's small, uncharged, nonpolar molecule, so it will diffuse across. And for each molecule, it's combined with ribulose 1,5-bisphosphate in the reaction center of the rubisco enzyme, which brings them into close proximity, catalyzing that reaction. And then this 6-carbon molecule is produced or split into 2 3-carbon molecules, 3-phosphoglycerate. Okay, so again, similar process of breaking a bigger a molecule into its smaller components. And so these are what? Uphill or downhill reactions? These are downhill reactions. These are favorable reactions. Don't require the input of energy. So first step, this addition of ribulose 1,5-bisphosphate to CO2 to create a 6-carbon sugar, and then that's broken down into 3-carbon sugar, or a 3-carbon molecule. That reaction actually requires the combination of 3 of each of these molecules, and you get, so in one cycle you need 3 carbon dioxide molecules, which combine with 3 of the ribulose 1,5-bisphosphate, and you end up with the 6 of the 3 phosphoglycerate molecules. Here, the input of ATP, so there is a requirement for energy in the next step, because you're getting the phosphorylation of these 6 molecules. So ATP is donating a phosphate to the phosphoglycerate, so that it has at either end, the 1 and the 3 position, a phosphate group. And then an electron donor helps to reduce phosphate, leaving glyceraldehyde 3-phosphate. Now this is an important precursor for building very useful macromolecules in the cell. Okay, so things like glucose, more complex carbohydrates like starch. This is really the starting material. Also, fatty acids and amino acids use this as a precursor molecule.
Does that mean there are other sites in the cell that do this?
That do the Calvin cycle?
No, that create glyceraldehyde. Or is it always derived from food source?
Right, so in us it's from food, but in plants they make their own. So they're autotrophs, meaning self-feeders. Bottom of the food chain. They produce food for us. We consume plants as part of our energy that we get, part of those food molecules that we break down.
Not just energy, it's also substance that we need.
Mm-hmm. Right, so there are a lot of precursor molecules that are products, along with ATP, of cellular respiration, and those go into making all of the sugars, the fats, the amino acids that our cells need to make all the new bits and pieces of them. More membrane, DNA, etc., more proteins. Okay, and then the last steps of the cycle just regenerate ribulose 1,5-bisphosphate, which also requires an input of energy. Okay, so these are the dark reactions, or the Calvin cycle. So energy-consuming steps here. And the energy, of course, has just been generated in the light cycle. So they're really tightly coupled. You can't have really one without the other.